Genetics is the science of heredity. It includes the study of what genes are and how they carry information, and how we also study as to how these genes are replicated and passed on from one generation to the other between the organisms. And lately, a lot of emphasis is done on the expression of these genes. So this chapter will talk about microbial genetics. The first question we want to know is, what is the genetic material? The genetic material, as it's sometimes called as a genome, includes chromosomes and plastids or plasmids. Chromosomes are composed of DNA. DNA is the nucleotide base pair that you have studied earlier in other biology classes too, and these DNA, the nucleotide that pairs up the DNA can either be adenine, thymine, or cytosine, guanine. If you remember those from the other biology classes. Now, each nucleotide pair is linked up to form a long strand of chromosomes. So here in this particular image, this little structure in green over here is shown to be the ribose sugar, and the ribose sugar has a phosphate connected on one end, and on the other end it has a nitrogenous base, in particular called as the adenine for A, and this one is showing you the same thing, but it's got thymine. Now notice up here we have the two strands, as we call them, as anti-parallel. So we have the two strands over here as anti-parallel. The reason for that is, notice that the 5' end is on the top here and 3' is on the bottom here. On the anti-parallel strand, the 3' is on the top and the 5' is on the bottom. And also notice the orientation of these nitrogenous bases. These are each 5 carbon bases, and notice the pointed end, which is the 2 carbon, is facing down on this in particular end on the left, and on the right it is pointing to the top. So these are anti-parallel strands, each strand that is then held together in the middle by these adenine thymine bonds between the two nitrogenous bases. Now here is the same image shown to you in a little bit more depth, in which we can actually see that between adenine and thymine there are 2 dotted lines, and between guanine and cytosine there are 3 dotted lines. And that is the reason why adenine can only base pair with thymine and guanine can with cytosine, because there are 3 hydrogen bonds possible between guanine and cytosine. 3 hydrogen bonds, and if we look at adenine and thymine pairs, we have 2 hydrogen bonds here. The bacterial chromosome, on the other hand, has a very unique feature, that it is circular in region. It's not linear as the eukaryotic nucleotide is, where it is you have the linear 2 strands going back to back. So make sure you understand the term genetics. Genetics is mainly the study of heredity. Genes are the segments of DNA. In viruses, sometimes we also have RNA as the segment. And the word genetic code is based to the nucleotide sequence that is present, which will then guide the amino acid to be formed. So genetic code is always a 3-letter segment of the nucleotide, of the chromosomal nucleotide. So in other words, we can have, for example, you can have AAT, or you can have CCG, and these would all be genetic codes. Let me cut this out so you can see it better. There, that's better. So the genetic code is going to be 3-letter sequence of either the combination of adenine, thymine, guanine, and cytosine. And if you recall, there are 64 combinations possible with just 4 nucleotides. So the next question is, what is the purpose of DNA? DNA has the recipe for making a protein. Proteins have many diverse function within the cell. They can be enzymatic proteins, they can be structural proteins, they can be proteins for storage, there can be proteins that are for defense mechanism, there can be proteins for signals, certain hormones can be proteins. So we have many different functions of proteins. The manufacture of protein is the most important aspect of DNA. So here is DNA shown to you. DNA cannot form protein directly unless it forms RNA first. So, for example, DNA can form RNA and then form protein. Protein is then folded to a three-dimensional structure, and on which an enzyme can work, and we'll talk about enzyme substrate. We actually did do that previously. Enzymes, remember, work on certain substrates which can give us products. So this would be the combination or this would be the flow chart between DNA and protein. Now, what is genotype versus what is phenotype? Genotype of an organism is its genetic makeup. That is information that codes for a particular characteristic. So, for example, if you have brown hair, or you have black hair, or you have blue eyes or green eyes, those are all in your code. So that's a genotype. Now, phenotype refers to actual expression, such as the what is the for example. Let's look at, for example, a combination that I'm going to write over here. For example, we can see that capital T stands for tall person. So having a combination of capital T, small t, and then a combination of small t, small t. So here we have, if you remember from genetics, capital T are dominant. And over here in the second combination, we have capital T with a small t. And this is called as a heterozygous combination. And in the last one, we have the two small t's. Now, the expression of the capital T, which is the tall, will be expressed. So each of these combinations is actually shown to you as genotype. If I was to do a phenotype, then the combination for this one would be tall. This one would have the expression of being tall, and this organism may be short. So the phenotype is the expression, which would be here, and the genotype is the genetic makeup. So you can see over here the genetic makeup can be different, but sometimes it can have the same phenotype depending on what genetic code it is. In molecular term, an organism's genotype is pretty much the collection of genes within its entire DNA. And what constitutes the organism's phenotype is, it's in molecular term, is the collection of all the proteins. So most of a cell's properties are derived from the structure and function of its proteins. In microbe, most proteins are either enzymatic, that is, they catalyze particular reactions, or they can be structural. For example, proteins in the cell membrane or flagella. Even phenotypes that will have structural proteins are indirectly on proteins. So the connection between genotype and phenotype is always there. What is so special about bacterial DNA is that bacterial DNA is, firstly, as I mentioned earlier, and we learned earlier, it's circular. So notice over here we've got the circular DNA, and it's also attached to the plasma membrane at certain points. And the reason for that is there is no nucleus. So it is not present in an enclosed combination. It's sort of free floating in the cytoplasm, but attaches on the surface of the plasma membrane. So here we can see that if this is the parent DNA, and when it undergoes replication, it shows that there are two rings forming, and then both of these rings can be then passed on to the new daughter cells. So almost like if you have two bracelets and you slide each bracelet away, that's the combination that you see over here. How is DNA copied for replication? Recall from the DNA structure, we had a very complex combination with in which we had an enzyme called as a DNA helicase. What the purpose for that was to snip the two strands and keep them separate. And there was another enzyme called as DNA polymerase that copies at the replication fork. And if you recall, it always does it from the end 5 to 3. So you would have, if you remember, the leading strand is shown over here because the DNA can open up from 5 to 3 and it's going in the top direction. On the bottom strand, notice that you have the segments forming in pieces, and that is the lagging strand. The lagging strand were later glued by the enzyme called as the ligase, and each segment are the Okazaki fragments. So I know that I taught this very well in my biology classes, and most of you have done that. I want you to watch an animation on this that I have it on the web study on this week, and it pretty much shows the entire step as broken down into each individual pieces. So make sure you do watch this animation. We also call DNA replication as being conservative. The reason it's called as conservative is that when the DNA is replicating, notice the origin of replication in a prokaryote will be on one side, and then it starts to go on clockwise and anti-clockwise sides, and it pretty much makes a copy of it. And when it is distributed, both of them get one copy of each. So in other words, when the DNA strand opens up, there is one new daughter strand and one parent strand is left. I'm going to show it to you from the previous slides, for example, over here. So notice over here, in the top, we have the DNA double helical. And when it's opening up, one of the strand is the original strand, and based on the base pairing on the original strand, the daughter strand is being formed. The daughter strand is the one that is shown in darker blue over here. So this is a conservative, semi-conservative manner because the new cells, the new DNA strands will be one old, one new. If we had all old or all new, it would be either conservative or totally new. So it is semi-conservative because it keeps the original strand from parent to the next generation. What are the specifics of DNA replication? Again, this is referring to the same animation. Firstly, DNA strands are anti-parallel, as I told you. Notice over here we've got a 5' end here and a 3' end here, and then on the other strand we have a 3 and a 5. So the DNA always starts at the new 5' end. So the open 5' end is the strand where the new strand is going to be forming. So notice the dark one over here is the new strand that is forming. Now as the zipper unzips, one end will always have the 5', so it is the leading strand. On the other hand, because it can only replicate from 5 to 3, it has to wait till it reaches a certain segment of replication, and then the RNA primer starts. But on this case, it's now starting from the inside of the replication fork and moving outwards, and as this will take place, it will form segments from small segments, and as the DNA opens up further, one of the leading strand will be continuously formed. The lagging strand will again be formed in segments. Each segment is called as Okazaki fragments. The Okazaki fragments start with an RNA primer, which are later converted to a DNA nucleotide. There can be certain errors in rotation, but a very, very slight chance. So I want you to watch this animation on DNA replication on web study, and make sure you understand each portion of this.